all back on board because we will have now the next session by Martin Seeber, who is going to look into analysis approaches of combined EEG and motion capture data with a specific focus on Gait here. You've seen great things he has been doing previously in his papers on the talk on the first day. And today he's going to talk a little bit about how to do that. So thank you very much, Martin, for joining us here. And the stage is yours.
Today I'm going to talk about EEG and Gait analysis, where I try to combine EEG analysis with the behavioral data Uros and Betty were beautifully introducing before, and trying not to only look at either EEG or the behavioral data or motion tracking data, but trying to combine that to some extent in a very simple way. So the aim of this session is somehow linking these two worlds, behavioral science or science that is more based on behavior and neuroscience and or EEG analysis. So in the to go back to the overview, we already have a long journey behind us, coming from the really raw data to the pre-processing and motion processing, and today we're almost at the end of our journey today. So meaning within this journey, I'm on rather the end, and that's why I'm in the happy position that I'm able to use the previous results already. I have really some very nice data because the hardest job, so to say, is already done, meaning synchronizing motion tracking plus EEG and pre-processing them in a way that they are synchronous. And in terms of tools, today I'm going to try to focus mostly using EEG Lab because that is a very strong toolbox in the community, but also for visualization purposes, I'm going to use Brainstorm to introduce and talk about source imaging, distributed source imaging. Today I chose this toolbox because it's open source and it's widely used in the imaging community. Personally, in Geneva, we have Cartool, that is cartography, a tool for brain topography initially. That is what we usually use in Geneva. If somebody's interested in that toolbox, please check it out. But for now, I'm gonna use the top two ones because they are more widely used, I think, in this community. And I think I just wanted to contribute in something that is already used and known by people here. So in terms of the program of today, there are three parts. First, I try to focus on comparing conditions. With that, it's just very simple comparison between what is the difference between standing and walking and what are the neural correlates of that. And on the second part, I want to focus on gait phase-related EEG analysis. I'm trying to use the markers that were provided by Betty and Uros to reconstruct the gait cycle based on behavioral and motion tracking data, and then use this behavioral data to analyze the EEG in a certain way and to establish a link between this gait phase and the gait cycle to brain dynamics. And in the third part, we're going to talk about distributed source imaging in that context, meaning the first two parts I intended to be very, very simple. So for now, they're very simple scripts provided for you, and they should show you the phenomenon you're studying. Meaning for the first two parts, I stay on the scalp level while I only go to the source level. But I already try to squeeze out the relevant information and show you the relevant phenomenon already in the first two parts using as simple analysis as possible. And in terms of data and scripts, as already introduced by Zane, we're using folder 5 containing the EEG and the folder 6 containing the motion tracking analysis or motion tracking data. And in terms of scripts, which are able to replicate the results I'm showing today, you can try to replicate yourself using the first function that is called compare conditions that is made for the first part. and then the second script that is made for the second part. Meaning all results from the first part should be replicable using the first script, while the second script is for the second part. And the third part, they're going to do a live demo, and that's somehow also included in the second script. So that should all be nicely provided for you and hopefully usable and runnable. After setting the paths, and as indicated before, it's very similar to the previous workshops. Just before diving into the data and into scripting and heavy data analysis, I want to highlight one more point on a more general topic, meaning when we study brain dynamics, or if you want to study the brain in general, it's always quite important, and I agree with those authors, to include behavior or to consider behavior in our neuroscientific investigations. And especially for the younger colleagues here, if they don't know or they haven't read this paper yet, I highly recommend reading that paper either tonight, over the weekend, or next week at latest, because I think it's very, very important paper in general when somebody is either interested in neuroscience or behavior. And I think it's very nice discussion going on in this paper, how to find something out about the brain and not trying to get lost on the way. Because we all know in this community, it's not easy to somehow bring these two words together, neuroscience and motion analysis, or MoBI in general. So we have to do some heavy processing. But I think what we never should forget is what is the purpose? What do we try to find out with our hacking, with our analysis? And for that, I think this paper is beautiful to read, and I even recommend reading it. So in that spirit, so to say, what I'm trying to do here is, in this first and second part, trying just to highlight what is the difference between the first and second part. Well, if you sit down with a colleague and just talk about, well, I mean, what is Gait? And if somebody would not know that, one could say, Gait is a certain kind of movement, right? You move, and the contrast to it is you don't move, you stand still or you sit. So that is just a simple contrast, meaning a binary comparison. And that is what I mean with part first. What I call in my papers usually movement state related activities, because you just compare the state of pre-movement or non-movement in contrast to the movement, right? If you would think of a regressor model, it's just a binary comparison being 0 and 1, and that is then just the contrast. That is the simple thing we can do, just comparing what is the difference of any movement, including Gait and being still. And the second type of activities we can envision are movement phase related, or in this case here, Gait phase related modulations. Because if you continue the discussion with your friend, talking just conceptually what Gait might be, one could say, well, I mean, Gait, you move your legs repetitively, and therefore there is some kind of phasic activity you can study. Meaning, in addition to this movement state, within this state, there are different cycles, right? Gait cycles. And if you envision that this is some kind of a movement trajectory, you could say, okay, those are like 5 parts, and they are repetitive, so I split up all these 5 parts, and those are kind of the same thing. So I try to get to study the brain activities as a function of these cyclic activities, right? And that's already conceptually, if you don't know anything about the brain at all, that's already only conceptually. If you discuss with a friend what Gait might be about, you already can figure out these 2 different phenomena. Meaning you can just look at the state related or contrasting information versus this Gait phase related activities. And with Gait phase related activities, one could have the hypothesis, okay, if you have these Gait cycles and Gait phases, then we reconstructed what we might expect. What we might expect is that the oscillation, certain frequency, is then co-modulating within this Gait set. I try to replicate these results today with you. So even looking at previous literature, this is really ancient data here. What is called classically in the literature event-related desynchronization or synchronization is exactly this almost binary comparison, meaning we normalize the power in a certain frequency band in respective to a pre-movement baseline. Meaning we have, in this case, we have a certain oscillation that is 10 or 20 hertz, the moving beta rhythm. And as soon as this participant was moving their legs, then there is this, these oscillations, they vanish. And if you then would compute the envelope of these oscillations, this is displayed here. First, not much is going on by definition, because that's the baseline. As soon, even before moving, you can see this suppression of moving beta rhythm. That is a very prominent and ancient feature we know for a long time, and that is something we see in humans, but that we also see in all kinds of species in animals. So let's dive right into it. As I said, and for everybody who was attending my talk at the first day, this is exactly what we found. We tried to separate movement state from movement phase related activities, or Gait phase related activities, meaning if you just compare standing to walking, what we see, if you look at the Gait cycle, there is this huge blue activities indicating suppression, right? It's just a negative relation between the power in the standing versus the walking condition. And that's present for the whole time. And then if you try to subtract everything that is permanent, we see a different feature here coming up that we call the Gait phase related modulations. And this is the difference between part one, where part one is this difference here. And part two in my presentation is trying to get those features. Good. So far, so good. That is somehow the aim of these the 2 days lecture or workshop, is to try to replicate those data that is already quite, has some years already experienced with data you shared with me, and that is not my data, and I don't use the exact same methods as before. It was recorded by somebody else and with different equipment. But just for sake of transparency, and I try to replicate those findings we had before, because it's nice we have a clear clue what we try to find and replicate, and it should also ensure us that we could replicate previous results. And also we want to introduce what I used before as artifact correction method, meaning trying to disentangle different spectral profiles in order to do some noise correction. This is mostly correcting for muscular needs. So those are the more or less three things I try to do, is trying to replicate this beta suppression and gamma modulation, and trying to introduce how you can do this. And in the very end, I'm going to talk about source imaging and going to the source space. Good. Let's dive right into it. Just focusing on the script, you can open up the first script that is called Gait EEG compare conditions. And in the very top of the function, there are all the settings, meaning in that sections, you can set your path, the file name. File name you should not change, you should not change, but the file path you just change to the directory of where you were downloading this folder file, right, for now. And the rest should be automatic. What is very important in this presentation is here also is to select the bad ICs. Because we beautifully learned from Marius and Anna yesterday that how to analyze and come up with these IC topographies and how to interpret them. And for now, I'm just looking I was just looking at these different components, and we're selecting only a few of them. And we discussed that in the workshop already. there are somehow two different approaches in the field. One is to ignore the bad ICs, and the other ones is just looking at the good ones. But in the outcome, that's a very different approach, because if you just ignore the bad ones, usually I use as a rule of thumb not to reject more than 10 to 15% of all the ICs if I do a back projection. If I just look on ICs that are good, the dimensionality is much lower, but maybe the data quality. So there is already, you can see the discrepancy of these two different approaches, where I'm just in the base camp of trying to reject things we're sure or we're pretty certain that are right. So that is what I did. For now, I just corrected 4, 6, 7. No, 6 components, meaning 1, 2, 3, 4, because these are clear IR artifacts. Those are clear IF artifacts. I think those are neck muscles as well. Those are 5 seem to be a brain component, so I didn't reject this one. And then I also looked at the component 8, looking also not only at the topography, but also time course and the spectrogram. And I just concluded not to reject too much. So I just came up, okay, let's do the worst components I know in this data set and try to get something meaningful afterwards. And I think the essential difference between these two ways of using ICA is either doing back projection or not. But if you do back projection, I think you have to aim for a high dimensionality of your data. And I think Marius was talking a lot about the rank of the data. That's exactly what we want to have. We want to have a rank of the data that is somehow decent if you do a back projection. If you look at simple components, that's completely something else. That's by definition rank 1. And that is just hopefully a very clear signal. So that's somehow the difference of the different schools of using how to use ICA. I'm just trying to keep it simple, and for now I just reject everything that I think clearly is an artifact. Good. First of all, we try to load the data here and just plot it. And I think also before doing anything with data, especially if you didn't record yourself, it's important to look at it. So if you just load the data here as indicated, I just load it and plot it. That's it. And what you can see here is, I think, 20 seconds of EG, and it's quite nice recordings, but I mean, there's also quite some noise in it. And those guys here, they're just I artifacts, very, very clear I artifacts. And when selecting these components, as I did it, we can try to get rid of those, meaning this is exactly the same scale, same data, same time range. And if you go back and forth, you already can see that the ICA correction did a good job. It's not perfect data because there's still some noise in it, but it's, I mean, it's way better. Just compare the former to the latter. This is ensuring. And also would like to suggest, if you look or if you load data or get some data, or if you record yourself, I personally, after I think 10 years of EEG analysis, still scroll through the whole file every time. So meaning you can't be, I think you can't be experienced enough for not looking at your data. I think really, I mean, if you are a good coder, that's great. I try to be too, and you know, I mean, everybody, I mean, some of us are trained as an engineer or even computer science. I am too. But still, this is what you do. You look at the data, and you should. I think you should look at the data after every computational step. And that is really checking if your algorithms were good. What you should not do is fiddle around with the algorithm, checking the data and fiddle around, but rather trying to get an idea what's going on and try to stick to the same settings and then go ahead. And what I also would suggest to do, still, that's a bit old-fashioned viewpoint, but I'm still doing that, is scrolling the data and marking the bad box. Meaning, if you have already the segments indicated or defined that you use as your EG of interest, you can still, even after automatic pre-processing, check if there's something remaining fishy things in your data set. That's a bit subjective, but I mean, I want to use an analogy of a colleague here in Switzerland. And he said, you know, cleaning EG is a bit making like a salad. You know what you not include in a salad, right? You probably would not eat a snail. You probably put out the brown leaves. So everything that is not appealing to you, you try to reject. And I think it's kind of a good analogy. And I think it's important to learn from your more experienced colleagues in your lab. If you don't have any, if you're the only person in the lab analyzing EG, I would suggest taking online tutorials and then still trying to get an idea of how to analyze EG, because I mean, EG is basically analyzed now for 100 years. There are a lot of people knowing how to do that. And there's really quite some tradition. It's really always changing fast. And now we have all these great computational tools. But I think the main points that were, you know, told, they were used for teaching 20 years ago or 30 years ago are still good to know. So meaning we try to come from raw data or pre-processed data, including artifacts, to more clean EG data. And that I just did using a filter, very simple Hypers filter. I was setting here on the very top of the function at 1 Hertz. I filtered the data. I tried to define also in the settings what are bad ICs, and then I said everything is good except the bad ones. And then here I just do the IC back and forward and back projection, and then in the end I end up with this clean EG data here. It's a very standard procedure. And the second thing I did, because for sake of time and making everything automatic for the course, instead of looking through the whole data set, which I did, but I think we can't do that now and not exactly the same way. I did simple thresholding, meaning I'm looking for epochs in the data which exceeds a certain threshold. And that threshold I also was setting in the very beginning. For now, I was setting this threshold at 100 microvolts. And here I just do a very simple, instead of manually rejecting epochs, I still would suggest doing, I just did very simple epoching and wrote a variable that contains ones every time the data is good, with zeros every time this threshold is exceeded, plus minus a margin. And that is something I always do, in addition of manual marking the bad epochs. So for now, and also for the sake of making everything automatic, I did this. And now there is the important part by comparing conditions, because if you go very, very to the back, what we do here is comparing different frequency bands, so different the power for different frequencies in the data. And doing one way to do that is just to compute the power spectral density in both conditions, and then compute the relation. That's what I did here, and it's a bit of a busy slide. I have to apologize for that. So what I'm doing here is looking for the events where the standing starts and ends, and then I can find out, okay, there are 4 blocks of standing. And I just initialize all the parameters I need for the FP analysis. And what I'm running here is just only if these start and end events are matching, because there must be 4 starts and 4 ends. Then I start initializing this routine. And for every block, I compute a PSD. And these PSDs are then, from every block, are averaged to get the average PSD of one condition, that is here the power spectral density of the standing condition. And the reason why it's a bit so complicated, and it's not just, you know, p-Welch or, you know, the power of using Welch's method, using the range of this block, is because we have these thresholded bad events, right? So if you would just do a power spectral density analysis, including these bad epochs, that doesn't help. And if you cut them out, you have jumps in your frequency. That's not good. So what I did here is to try to get chunks of a specific window sizes, do the frequency analysis for every window, but only for all windows containing only good data. And that is what I'm doing here. Basically, it should be as similar as possible to the power spectral density computation as well, measured methods, without the bad epochs. That is quite helpful. I can do that for all 4 blocks in standing after, looking up where is standing, which are the blocks, which are the chunks, and then summing up all these blocks. Having all the blocks, I can average then all blocks together and call that power spectral density standing. I can do the exact same thing, and the code is exactly the same for the walking epoch. And here it's just the difference that I look for where walking is starting and walking is ending. And then I do exactly the same thing and come up in the very end with a variable that is called PSD walk. So it's very nice. I have already the power spectral density computed for standing and walking. And now I can compare. I can plot those. So this is a plot. I do that for every channel, of course. This is a plot for CZ because we know this is the most interesting electrodes given the previous literature. And I can just plot the power spectral density in microvolts square per hertz for all the frequencies. So you can see this nice 1/f decay, and you already can see that during standing, there might be higher beta activities here, and during walking, there might be this broadband activity that is very strong for frequencies above 40 hertz. And here I didn't do any sophisticated cleaning. That's why I think this is very stereotypical for macular artifacts, and I'm going to talk about this in more detail later. But it's already quite nice because we know it's kind of the same thing, but there are some differences. That's exactly what we expect. Meaning the overall shape, meaning we have a 1/f shape, is the same, and but there are still some differences we think it's worthy to analyze further on. So what we can do with those guys, we just have the PSD walking and the PSD standing. We can just compute the difference of it, so the log difference, that is the power ratio in a log scale, and call that ERSPs because it's one snapshot of ERSP. And then trying to plot that. In a second, also, I'm going to explain how I do this PSEA, meaning principal spectral component analysis denoising, I call it, removing this broadband component. So the idea of that is more or less we have a representation of these already computed without any correction so far, meaning we have some physiological activity, plus we have some broadband noise. And what is the aim of the principal spectral component is trying to find a spectral component that is very prominent and has a spectral shape that is indicating muscular activities. And for muscular activities, we know that frequencies, the power for frequencies below, let's say around, below 20 hertz is low, but for higher frequencies is high and it's broadband. That's exactly the spectral profile we were showing here in this paper, meaning yellow means low power, red means high power, and that's quite consistent across participants, but also across time. It's very stereotypical, meaning the uncorrected data looks like this in time, frequency, and space. And if you look at always the first principal spectral component and remove that, meaning this minus this results in that, we have a much nicer time frequency plot that is not that much affected by these artifactual activities. Plus, even more strikingly, if you look at then the topography or source images, those guys should be gone, meaning these plots, meaning per definition, these activities plus these activities result in the raw data, but what I did is the opposite. I just get the raw uncorrected data. I want to split it up in things I think they are physiological and things I think they are. And that works pretty well in a lot of people. So computationally, for the nerds, I use this USP variable and compute the covariance. So this is already a normalized. This is not PSD anymore. This is already a normalized matrix. I compute the covariance in space. So what I have is in the end is a matrix that is the number of frequency bins times the number of frequency bins. I do eigenvalue decomposition here, and this is I do a PCA basically using eigenvalue decomposition. And what I find is then just principal spectral components that define the space I was recording before or was getting before. And I can split up those spectral components, and always the first one is the bad one because it's the strongest one. And then I just project the first one in a variable that is called PC1. And all the rest, meaning all spectral components from the second until the end one, I project in a variable that is called ERSP corrected. And then that should be this, right? So the PSC1 should then be the artifactual activity, and the PSC2 to the end is the PSC2 to 100 here. Because here I had 100, here I just have, I think, 64 channels. And if you do that, it's very simple, just some lines. If you do that, we can look at the ERSPs coming from these raw power spectral densities. First, we just do a comparison, log comparison. And then we can do this log comparison plus the correction. Meaning, if you do the raw one, you already can see for frequencies at around 20 Hertz at CZ, we have this suppression. You say, see that here is 0. Plus 3 would be a positive, meaning power increase for a certain frequency band. And minus 3 would be a decrease at a certain frequency band. What we can see in this data set here, just one subject as an example. It's very nice, somehow matching to previous results. What we see is a suppression at around 10 Hertz, but a much bigger suppression at around 20 Hertz, what is nicely matching to previous results. And if you do a decomposition of this spectral profile, because you already can see this huge offset here, that's around 2 dB, that's almost constant for frequencies over 30 Hertz. You can do this PSCA. And here I plotted also the first principal spectral component in that analysis, and that is the red trace here. Meaning, again, we know that this trace, the first trace, the raw trace, is a summation of artifacts plus physiological activities. So if we then get rid of these broadband activities over here that are low for low frequencies, but strong for higher frequencies, and that is very characteristic for muscle artifacts, we can then come up with the corrected power spectral density plotted here. And here we have a bit a stronger effect, plus there is still a bit an offset, but this offset in power contamination for higher frequencies is just very much reduced. Does that make sense? In terms of topographies, this is what I showed here is just one electrode so far. It's just CZ so far, but I can do that for all electrodes. And this is exactly also what I do. So if you do all electrodes, what I'm showing here, this is not voltage here. This is power ratios, right? This is the ERSPs as shown as topographies, meaning this is not the same as what we look at when we look at ICA patterns, because there we look at the voltages. Here we look at a color-coded representation of the power ratio, these ERSP measures. And that we can do for all channels, for all frequencies. It's exactly what I did. I scrolled through and plot for 2 Hertz, for 4, 6, 8, and so on. And that is already quite indicative what is going on. And what is very nice to see already in this data set, that is your data, is we saw suppression at around 10 Hertz over visual parietal areas that is very characteristic. But in addition, for beta ranges, there is the second blob coming. This is not yet source analysis, but we still see the phenomenon we're interested in. And if you take these uncorrected topographies, we try to split it up as described before. And if you do that, those phenomena get stronger. and those guys are vanishing. So if you look at the topographies for these higher frequencies over here, starting at 30 Hertz, this is more or less always the same picture, right? And it's always the same picture because it's really averaged data. And I think it's mass selectivity. And if it's really broadband and affecting somehow always the same areas, that should be the same. That should be the same topography, right? So meaning it's kind of a boring thing because it's broadband. It's a contamination, so to say, and it's always more or less the same. It's not frequency specific. And if you correct that, it's still there's still some noise because I didn't do too much cleaning before. But these guys are gone. So we start with the raw ones, and we can split up these raw topographies in two parts. That is, so this is the raw ones. These are the corrected ones, meaning the good ones. And this is the topography frequency dependent for this bad component. And it's per definition always the same because it's always the same pattern. Right? So if you look at the raw data, we see, okay, it's the same, and then we just remove one specific pattern, and then it's just, of course, always the same, but it varies in strength. Meaning this plus this would then result in the original data as well. And that's already the end of the first part. Meaning we can look at the USPs in terms of frequency spectra. We found this very strong beta suppression that is located over sensomotor areas roughly. This is just a bit speculative for now. But also we were successful in replicating the spectral shape of this artifactual component I was removing before. And it's actually, if you move and compare standing to walking, you always can find, at least I found that in every participant I was studying to. And to set that into context, this is the current data set where I show mutant beta suppression somewhat over central areas. That is quite matching the previous results showing that central sensomotor beta suppression, meaning this spectral shape, especially the suppression here at 20, that should match the suppression here at 20 I showed in my Frontiers paper, plus the spectral shape I was showing in the data set I did in my PhD. There's a spectral profile for the artifacts being like this, very characteristic, and that's very similar to the one we showed here. And if you compare then those two in the central sensomotor regions, this is very, very similar to this is in the paper, and this is exactly what we can reproduce in the data set you shared. So that's already quite encouraging because we can kind of see the same phenomenon showing very similar characteristics. This is quite ensured. Maybe before we go to the second part, maybe we can try to discuss some questions if there are any already, just to chunk it up.
I don't have direct questions in the Zoom chat nor the Discord channel, but one quick question from me. So the order that you showed here was clearly ICA, remove I components, and then do your frequency decomposition. What would happen if you did that before ICA?
The PSCA?
Yeah.
Yeah, that's an excellent question. I think it won't be so nice because the ICs are really affecting. Especially the I blinks are really changing a lot, although they are in lower frequencies. But I think the reason why it's quite robust is because there's the averaging before, you know. I think I was working on trying to push that method into a pre-processing method, but then already noticed during my postdoc, this is kind of opening Pandora's box. So I had to decide if I go on with developing an artifact correction method, and I was very aware this would take me 1 year, maybe more, or if I'm more interested in finding out what the brain is doing, and I was going for the second option. So I'm happy to have younger colleagues that was maybe still on the beginning of the PhD, and I was even expecting somebody maybe taking that over. I'm happy to share all knowledge and functions, But for now, I think, yeah, I'm out of the stage. I still can do that, I mean, for sake of time, unfortunately. But I mean, I tried and I played around with it, but I mean, it's then just in the details. To make something automatic, you can distribute. For that, is too premature to have something, you know, because it's PCA based, that could be online. It's real-time capable. And there is one alpha version I wrote that was promising, but then I really had to stop because
yeah. Okay. Thank you very much. So are there other questions to talk up to this point?
Yes, I do have a question, as usual.
Yeah,
Thank you very much, Martin, for the nice presentation. I was just want to get some elaboration on this picture you have on the left dorsolateral, left and right dorsolateral. Are they? What is the message here?
yeah, I think this is really an overlay. You know, when you just look at 12 hertz, it's only on posterior areas, maybe left, maybe right. And then there's a third patch somehow coming in. And I think this is just the overlay of different sources with different frequencies. I think that is, I mean, I don't have a functional interpretation for now. I think if you want to come or work towards a functional interpretation, you have to address it experimentally, meaning you could say, okay, maybe there is like left and right posterior activities, and that might have following function. Then you have to decide an experiment trying to answer the question how that feature would be behaving in certain paradigm. Right. So for now, this is just correlation.
Yeah, yeah. Thank you. And another question in the IC components in the beginning that you were showing, there is one component, I think it was component 19.
Yes.
I was wondering, because what I learned is that this component in the center of the head is a very common noise that, yeah, exactly. And it's usually being removed. I was wondering why.
Oh, that's a very excellent observation. I mean, especially this component is a very interesting one, because I mean, here first it says brain 82%, and second it's over central sensory areas that could be lag activities. But it might be also activities from the cheeks, right? That might be a mix. But I would rather say this is a brain component, but I'm not an expert in interpreting that.
Yeah, in terms of mathematical and calculation, is what I understood is that,
yes.
Based on that, it always there is this component that is in the center of the head and needs to be removed. But
right,
yeah, I don't.
Oh. Well, I would not necessarily agree, okay, because it could be physiological activity, and that's exactly why I don't.
Yeah,
what is a good example, I think, related to your example, if you have two dipoles in the auditory cortex, that sums up also to a central prefrontal one, or only the topography, meaning we have to interpret the topography with great care. It's not given that the sources of it, the sources of the topography you can guess only by looking at on these contour lines. With that, you have to do sourcing. I think we can have a somehow qualified guess, but in the end, we have to be careful looking at sources, but also always considering the context of our experience. I think that's very important. For this component especially, I would not reject it only because it's like this.
Yeah.
That's somehow a bit on the conservative side. I kick out everything that I'm quite confident it's not. It's not. And here, I think I'm also quite sure that is some kind of brain activity. That's always kind of the thing, right? There might be some brain activity in most of noise, but even if you remove something that contains brain activity, even if it's just a little, you always lose a bit, right? That's somehow the trade-off. And we don't have a clear answer for now, but this is what I'm doing.
Yeah, that's true. Yeah. Yeah, thank you.
Mars.
Good question.
How do I pronounce your name correctly? Marcy?
It's Marzieh.
Marzieh. Yeah. Could you elaborate a bit on the assumption that you made that there is a mathematical explanation why this should be an artifact?
Yeah, I by heart I don't remember. It's a very long time ago. I was playing with this with the data, and I look at the literatures and the way that they are removing in the Gait papers, and also what I heard is that, for example, for the IC or PCA. Based on this, because the head is like look like a sphere, and the center, it's always like I can't remember or say it correctly now, but yeah, based on what I understood is based on the calculation, is at the center. It's always get the most weight. And it will be represented on the topography in the center of the head. It's yeah, what I can remember for now. If I say it completely correctly.
If you found a paper like that to post it.
Yeah, there. Yeah, I will. Yeah, I will post it.
So from our experience, I would entirely agree with Martin. So I would double check, of course, like all the other dimensions to see what this might be. But we often encounter components with such a topography that are actually on sensory motor cortex.
Yeah, I was also very confused in the beginning when I saw that. But yeah, maybe I was still wrong. But yeah, I will send it to you.
Thank you very much.
Yeah.
Okay, I think the question from Hannah is answered. Is that correct? Yes. So Martin answered Hannah's question directly in the chat. And is there any other question? Okay. So okay, so there doesn't seem to be any more.
Sorry, maybe I have a question. So you just showed that you just remove like the first of the PCA components. So is that like a common thing that you just saw that that's enough, or do you look at the others as well, or like, yeah, I just wondered.
I mean, I look at all, but what is enough, we don't know. Right? Even you discussed that. I orient myself usually at the first ones because what I'm trying to do mostly is removing the eyes. That's the most important thing. So after removing the eyes, and if I find a heartbeat and get rid of those, I then try to find some muscle artifacts that are strong. And as I got it correct, those components are sorted by variance, meaning those are the strongest ones, right? So what I'm trying to do is trying to find the eye components, the heart component, and the worst of all muscle activities, and then I cross fingers and try to mark the bad box and try to get this.
No, I actually was talking about the PCA. So because there you plotted the first one, and then you plotted 2 to 100, and like I wondered how the second one would look like, and if that's just another offset of the muscle, for example, or what that would do.
I mean, first it's spectral components. It's different because the dimensionality is the opposite, because here the varying dimension is the time dimension. In my case, the varying dimension is the space. So we get spectral. So what you got to get here is spatial filters that has the dimension of channels. I get spectral filters having the dimension of the frequency bins. Meaning if I have 100 frequency bins, those are 100. You know, dimensionality of 100, meaning what these patterns would be, in that would rather be the spectral profiles. Only the back projection then gives us a pattern, but they are less meaningful because there are eigenvectors, and we discussed already that the eigenvectors or PCs, they're not physiologically meaningful that much. Meaning the first one we can be mostly sure. It's also how we discussed that the first one might be meaningful, but the second one might not.
Oh, okay.
While in ICA that is the case. Because per definition, usually the first principal component is always the smoothest one. And here is the smoothest one in spectral domain. And if you go to the higher ones, it's more noisy, it's more crazy. Right? If you try to get the summation of all your spectral profile, You have to have all of them. But the first one, I think it's the reason why it works out is because it's so strong, because the muscles are so much stronger than brain oscillations. First reason. Second reason, there is also this variation in space that helps us. And that's why I don't go for the second or third one, because that's already quite some dangerous thing. Also part of the reason why I stopped then developing, because I know if you go for overground walking, especially if there are obstacles, what we did here in Lausanne, this correction is good, but it's not sufficient. Meaning we do not get as great results using only, correcting only the first component, but we already knew we have to remove more. But then there is this question how to separate it. Then I was already, this is really now going a bit far, and I don't have a solution. But the first one always works out. So far,
so would it make sense to do it like twice, and to
no, I mean, if you do it twice,
the
first one is the second one.
Okay.
I think also mathematically that is the case.
Yeah, I'm not a mathematic.
Yeah, me neither, but I think that is what I learned.
Yeah.
It's also what I think the data shows.
Thank you.
Is that okay? Okay. Happy to answer.
Okay. So are there more questions? If not, Martin, will you go ahead directly, or do you
yeah. I mean, if everybody's still on board, I would go ahead and maybe make a break after the second part, because the second part is not as long as the first one, because the first one includes already, the main approaches I'm taking. The second one is just like an appendix of the first one, although it's conceptually very different. Okay. But the pre-processing and the general ideas are the same. If you agree, I go on for like maybe 20 minutes or so, and then we're going to make it. Good. Well, having already talked about pre-motion and motion or different conditions, this is what we did by now. I was averaging everything what is going on during movements and everything what is going on during standing or sitting. In this case, standing, to be precise. And then just compare that. Just did a simple comparison. Why now I want to focus on these cyclic activities, meaning we just not only do a binary comparison, but we try to find out activities that are co-modulating in the Gait cycle. That's exactly the topic of the second part, where I'm focusing on Gait phase-related activities. There is another script you can try to run. For the second script, it's important to also set the brainstorm path. The only function I'm using from brainstorm here is time frequency analysis. And that's the general difference. We discussed it already. We looked at those results, but this is conceptually very different, looking at different phases, meaning there might be a shift in the activities that is general during movement, but on the plateau of that state, there is within modulation that might be completely different from the first one, and that's exactly what we showed in the previous papers, meaning we do not look at this suppression or increase in power, but we look at the co-modulation. That's the simplest way of including the rich behavior we can track. And I have to say I was very impressed of Auro's and Betty's talk and how rich only the behavioral data might be and could be. Although Gait is very simple behavior, we can derive so many stats from it. But when looking at the brain, we just don't do that. And we just, I think, on the beginning, in trying to making this bridge to the behavioral scientists and trying to get a better idea how that could guide how that research could guide our brain analysis. So what I did is again setting the parameters, meaning you have to change the path to your EG lab folder, you have to change the path to your brainstorm folder, you have to change the path to your EG plus motion capture folder, the former being folder 5, the latter being folder 6, and the rest should be automatic. I want to explain which kind of components again I was selecting, how I did the thresholding, But basically, I did the same thing here. I just looked at the different subjects just to have some variance in the presentation. Again, I did a threshold of 100 microvolts and a high-pass filter of 1 Hertz. There is one parameter I'm going to dive into later. There is a tolerance of around 20%. Again, I'm loading the data, not only the EEG, I'm also loading the motion capture data. I apply a high-pass filter, and I select bad ICs as described before. This is exactly all the same. I use the same procedure with a different subject. Then I do the thresholding, again, looking at periods that exceed a specific threshold, meaning 100 microvolts, that is not plausible. And I mark everything that exceeds this threshold as being bad. If you look at the behavior here, just plot the position of the right ankle, similar as Uros did before. This is a trace of the whole experiment, meaning they were walking before, and then they were standing afterwards. And if you zoom in, you can see this back and forth walking patterns, meaning they walk they walk in space, but they do not only walk back and forth, but the movement is of course consistent out of different phases, meaning standing and swing phase, stance and swing phase. And having these stance and swing phases, you get a clue already when they did the swing of the foot. And based on those guys, this was provided nicely by Uros. We can define in these walking patterns, we can define the moment of the gait cycle. Meaning if we have the gait cycle plotted here along with these raw motion capture data, we can define gait cycles that are just here defined from 0 until 100%. And those guys, they can vary in length, meaning the steepness of those traces, they can vary. And the steepness of those traces, they are a rough indicator how fast you're moving. And also if you have different step length, if you just have these gait cycle periods interpolated, so to say, they can slightly vary. They can be longer, or they can be shorter. That's very important for the EEG analysis, because if you average, everything has to be exactly the same timing. But here we don't have the same timing. Meaning, what do we do then if you have different gait cycle length? And that we can cope by interpolating, having one gait cycle defined from right heel strike to the next right heel strike, and make an interpolation. Okay, we know this is 0 here and this is 100 here. That must be then there must be just a linear evolving from 0 to 100. And depending how long or how short this is, then these signals are more steep or less steep, but it already gives us a good indication which phases the gait cycle is going through. And we can use the signal we derive from the motion capture in order to analyze the EEG data. And that's exactly what I did. I was loading the movement trajectories I'm looking for the left heel strike here. Oh, I think that's an error. It should mean right here. It's called ASL. That's a wrong screenshot. I'm sorry for that. And then based on these events, I define this. I'm trying to define these gait cycles and compute the intervals. And having these gait cycle markers, those are just markers in time. I try to interpolate from every marker to every other marker. And the way I'm doing it, I'm trying to find the range of each gait cycle when it starts and where it stops. And here I just did a very simple linear interpolation from every event to the next. And this interpolation then really corrects for how long this thing was. And then I do some checkups. I remove everything that is out of the range, meaning I was computing the median duration of each interval, and reject every gait cycle that is longer or shorter than 20% of the median duration. Meaning I have all kinds of gait cycle length, but maybe there was some wrong detections or some problems in the recordings and or the algorithm. And then I just keep everything that is somehow within a specific margin. And that's exactly this tolerance factor I was indicating in the very beginning. It's here. So all those that are exceeding or are below a specific threshold, I'm gonna ignore. Meaning I again set a variable that indicates what is valid and what's not. I ignore everything that is not. I compute these gate phases as shown here. And those gate phases, and that's very important, they have exactly the same amount of samples as the EEG record. And then I can just compute a very, very, very simple MRCPs from them. Meaning I look for every phase in the gate cycle where this phase is occurring. Meaning I look for all the zeros, all the ones, all the zeros, all the threes, and so on. But for now, it's just for every phase, let's say 2%. I look in this signal where in time there is 2% in the gate cycle. And then I just try to find out all these events having this specific phase. And then I just try to get all these indices and say, okay, when the gate phase is exactly those, I just average all the EEG at that time points. And so I can implement something varying that is repetitive, meaning because 2 is not always at the same position in time, but relatively in the gate cycle, I can come up with a measure that is then just consistent over every cycle. That's the most simple thing you can do. And for now, I just averaged a gate cycle dependent MRCP. And this is exactly what I did. What you can see here is already quite suspicious. So if you just do that without any further correction, just remove some components and mark some valid box, what you come up with is these MRCPs. These are all channels. And what you already can see is these 2 very strong fluctuations here, meaning one is at about 0 and the other one is about 50%. But that is exactly the timing where the heel strikes are, meaning this is exactly when you are moving the head the most. And because we did not yet do a special consideration of artifacts, and these MRCPs are just containing, the raw recordings, no frequency analysis, nothing like it. I think so we should be most suspicious that those 2 huge fluctuations that are coming up at around 0 and 50% might not be physiological. To go on and to focus on frequency analysis, I just do that here for sake of time. For one channel, for sake of simplicity, what I'm doing here is defining the frequency axis, define the parameters of this frequency transform. This is the function I'm using for brainstorm. It's exactly the same approach, meaning looking at specific phases in the gate cycle and then trying to average the magnitudes. So if you do the Morley wavelet transform, you get a complex signal. If you use this complex signal that is time varying, it contains for this channel a time varying signal for all different frequencies. You just take the magnitude, you take the magnitude and you average all magnitudes being at exact the same phase in the gate cycle. And that you can do, but it's a bit a richer signal because there you have for every channel, you have a matrix that is number of time points times number of frequency bits. That's exactly what I did here. I did it for all frequencies between 2 and 200 frequency hertz, what is a bit too ambitious, I think. But what we found here is a modulation at around 30 hertz that is quite similar to the one we showed before, meaning in the gate cycle that is defined from 0 to 100% here. This corresponds to this gate pattern. And again, we find somewhat, it's a bit more noisy than in my previous presentations because it's overground walking, it's minimal cleaning, and a single subject. But it's quite similar to what we showed here, meaning here we can cut everything above 50 hertz, right? And if you would compare then everything that is below 50 hertz to the plot I'm showing here, this is just a more noisy representation or a noisy version of this gate phase related modulations me and Johanna were showing like 5 years ago or more. And that's quite encouraging because this phenomena already was showing to you. And that phenomena was just replicating, meaning, again, using different coding, different equipment, different lab, we can again replicate our previous results and having these different phenomena, meaning the beta suppression and the low gamma modulation. What I wanted to discuss and highlight that I believe that those guys here, again, broadband spread, I believe is artifacts. And this is all this as scalp analysis so far. It's not source, it's not hyper clean, but the phenomenon we discuss are the same as we showed before. And we can just add that. I mean, this is the slide I'm always showing for gait, finger tapping, circular arm movements, tapping your legs in the STN, bicycling, treadmill walking idea in Lausanne. You can just add this image now to, you know, the hallmark of replication studies we did. This is from the MoBI workshop 21. This is this one using your data set and the very, very simple pipeline I'll just share with you. And that is quite, you know, it's just like one band member of the club here. Right? This is just joining the gallery of gait phase related modulations we showed before, and also others showed. And that's, I think it's also quite encouraging that this hopefully is legit, and it just matches to our previous literature, but also to our colleagues replicating that. And I was quite happy seeing that it was also a bit a risky move to also be honest, that I was trying to replicate my results by data that I don't know. So, but it worked out, as why I'm happy, and it's a bit encouraging. So to sum up, what I was showing, try to replicate is this beta suppression plus the correction algorithm that is hopefully robust in those results. I have this beta suppression I tried to describe before, plus I have the low gamma modulation taken together. Those are two, as discussed in the very beginning, conceptually quite different features, because we already discussing with your friend conceptually can discriminate between different movement states versus different movement phases. And also we know, found out those guys are functionally different, because the core frequency again here is at about 20 Hertz, while the modulation is, I think, strongest at around 30 Hertz. And that is quite nice because it matches with your previous observations. And with that, I think we're gonna discuss some questions, maybe take a break, and then go on with sourcing.
Okay, thank you very much, Martin. Great. Super happy to see that you replicated that. Very nice. And thanks for the risky move. So, are there any questions?
This plot is only for one participant, right?
Yes,
Like,
it's only
yeah.
exemplary.
Okay.
What would be beautiful if somebody has time, I know you have the whole study, maybe Jana wants to do that, or one of you. You can analyze the whole study using that script. So we'd be happy to see the results. I think it's not every participant is as beautiful as that. Some are more noisy, I believe. But I think if you have like 20, that must be, like, I hope to have something like this here. I mean, to be fair, this is also one subject, but here I was more stringent with cleaning, you know, like manual cleaning in time, because here you can get fully automatic with minimal cleaning on the scalp, but it still somehow does the job. It gets better if you're more careful selecting bad components, not selecting better.
Okay. So I can't see questions. Please speak up if you have a question. And I'm sorry, I have to get out for 1 second. Zane, can you take over?
Oh, yes. Yeah, thanks a lot, Martin. Everything worked really smoothly and super cool code. Yeah, are there any questions from the audience? If so, then you can unmute yourself and feel free to ask. All right. Unless there isn't any more, well, we still have a lot of time. So is there any other points like?
Well, there's a third part. I mean.
Oh, sorry. Oh.
No worries. I just wanted to finish up this first and second part. I would suggest to do a break before going on to the third part, because I did not do distributed source imaging by now. It's just all in Skype, right? I just tried to focus on the same phenomenon we were trying to study before. What I want to try to do in the third part is introduce how to do distributed source imaging in a very simple way, and trying to load this data we've got here and try to introduce those ideas. That will take about, I think, 30 to 40 minutes from now. But I think before doing that, I would suggest doing a break, because 2 hours is already quite intensive, and maybe somebody wants to have a coffee or something. So I would suggest doing a break, I don't know, 10 or 15 minutes.
All right, then let's meet again in, say, at 1:20, so 20 past 1.
Well, that's perfect for me. I think the source imaging is not too long.
All right.
Okay. See you later.
See you at 1:20. Well, time flies when you're in a break. So let's wait a few more minutes, a couple more minutes, so that everyone has time to make coffee and stretch their arms and shoulders. Okay, Martin, are you back? We're having a few more minutes of break. Yeah, that break felt like 3 minutes to me.
Nice idea to rerun the data, Jana. We use like the standardized group just to see whether we can reproduce the results.
Yes, we should do that, especially for the both subgroups, and have a look.
Yeah, there might
And
be really informative changes in the better band during the dual task.
Yeah.
Compared to standing, that would be pretty cool. And age changes again, like in statistical interaction, would be cool. Significant.
Next on the list.
Yeah, maybe like a master thesis, because the data is pre-processed, everything is there. We do have the ICA. Martin can join us. Scripting through that would be a nice master thesis. So anybody out there who does not have yet a master thesis, send us a note. New master thesis in the making.
But we also have these condition. So the third condition, yes, with the vibrotactile warning is all in there too. So a lot of things we can do there with this data set.
Yeah, I agree. A lot of things you can do with a lot of MoBI data sets for years. Okay, Zane, are you still moderating?
It depends on if you are. So, hey, welcome back, Martin.
Hey, just did a coffee. Good.
So, shall we start now again with continue with part 3? Okay, great.
Ah, there's a question in the Discord chat.
Yes.
Yeah, thank you, Luisa. There is no script for this because it's very visual now. Everything what is computed I'm using, and the rest I'm just doing visualization. And the results then can be stored by clicking around in Brainstorm. Unfortunately, I didn't do that fully automatic. It's also a bit hard to compute something in MATLAB and then sneak it into the toolbox. That's why I'm staying within the toolbox. And maybe one side note on the results here. This is very simple comparison plus very simple gait cycle analysis. If you now think on the very rich behavioral data that Betty and Uroš were presenting before, you can think of, computing the same metrics, but then comparing different groups, meaning older and younger people, or trying to get these metrics and then trying to find out if either the modulation or the is dependent of whatever experimental condition you have and or behavioral metric you got. I think that is just a demonstration of what phenomenon we can observe. But if those phenomena are dependent on this and that experimental perturbation, that is actually the interesting question that it comes afterwards. And I'm happy to, if somebody would find time to do that or focus on this. And if you do, please tell me if you have results that are interesting. So for now, I want to go on with EEG source imaging. For that, I'm going to use the results that are produced by script 2. Meaning, if you run the second script, that is the gait phase related script, this is already, everything is already computed that we need. Brainstorm is an open source toolbox that is widely used in the community, I believe. If you want to learn how to use this toolbox, I can highly recommend either reading the tutorials, or they also do specific classes or tutorials themselves that are much longer. Now I try to squeeze in as much as I can in half an hour that just is related to our workshop. But of course, Brainstorm is much more general and much a wider framework that I think is still worth looking at. And I highly can suggest just to have a look. EEG Lab is amazing. I think you should use both. If you just want to have something some functions that might be not implemented in either the one or the other. There might be an option to have your function you need either in brainstorm or in EG. I think if you combine both, you have pretty much a lot. There's also M&E and other toolboxes, and everybody has his favorite tools. I'm quite agnostic to it as long as the results are well described and well done. I think that's the most important thing. Good. I'll try to switch. First, a bit coming back to the theory. For everybody who attended the talk in the first day, that it might be a repetition, but what we try to do is try to formulate a mathematical relationship between the source space and the Skype space, meaning we want to formulate a forward model that describes how the electrical potentials at every point in the brain might propagate through the skull. The only thing we can record is skull potentials. And from these skull potentials, and we already were looking at these topographies with the IC patterns, for instance, we want to find out which brain sources might produce the skull recordings, right? So the forward model is very well defined because there are more sources than scalp electrodes. And for just having a mathematical formulation, you can have as many sources as you want, and you still are precise in describing the scalp speed. But the reverse is not true. If you just have, let's say, 64 channels and you want to know 5,000 points in the brain, which might produce the scalp potential of 64 channels, then you have a problem in a mismatch in dimensionality, meaning you want to know more signals that you were recorded, right? And you also want to know the source of an effect, because actually the scalp potentials are the effect of the brain sources, but you just have the effect when you want to reconstruct the sources. That's why first it's very important to do the best you can do in forward modeling. I described that before. The first task is to co-register your electrode positions with the points in the brain you try to reconstruct your activity for. And then having these geometrical relations sorted out, you also have to insert and select specific values for different tissues in the brain, which lies between your brain sources and the scalp recordings. And if you have that done, this you can formulate mathematically, and this is beautifully implemented in the toolbox, how every point in the brain would spread to every electrode in the skull. And then you can take this relationship and try to invert that. But you cannot invert that very simply if you don't do additional assumptions. One assumption that is very prominent one could do, and I think that is very prominent assumption in the EG lab, if you have one dipolar source or one dipolar topography, you can go for dipole localization, because you know there might be one very strong dipole producing a certain configuration that is your IC topography. And we know that ICs were suggested to be dipolar, and if that all is fulfilled, I think dipole localization is absolutely beautiful, because what you have to learn there, you have 64 channels, and the only thing you learn is three coordinates and the strength of the dipole. Meaning you don't have to reconstruct a lot of parameters. You can compute that right away, the dipole from your topography in that case. If you want to go for these distributed source solutions, and I'm now focusing on this L2 family of distributed sources, those are all minimum norm solutions, then we try to get a distribution in space covering the whole brain. In that case, usually we have more sources than we have recording points. And for that, usually we do a smoothness constraint, meaning we make a constraint, a mathematical constraint, posing that neighboring sources should be similar. So we always get a smooth distribution. It's a bit washed out. That's why Loretta is called low resolution topography. What I did already in the second script or in the part 2, and I didn't tell you so far, I'm writing a specific structure for the MRCPs I was showing you before. I write a variable called mrcps.f, time and comment, and that contains the data, the timing, and a comment just having the name. So this is just the least you can write as a variable that is readable by brainstorm. And the same thing I did for the IC maps. So what we are looking at is these MRCPs and the IC parents, and then I want to do source imaging with those two guys. Oops, I have to thank my supervisors and funding agencies, but now I'm gonna go to the live demo, if I may. Do you see now a MATLAB screen? I think you do. Good. You, I think this is already done. I should have these variables in my space. Here is the MRCP, that's encouraging. And I just start now from scratch, brainstorm. I prepared something, but let's start from scratch. I really do. So this is the small window. This is the GUI. And when you start from scratch, let's really start from scratch. We make a new protocol. We call that MoBI live. And then we set these parameters. We want to have default anonymity because we don't have MR, and we want to have one channel per subject for now. So this is empty. This is just a name, no information so far. And then we can look what is already there at the presets. There is a patch here called anatomy, and another one that is called functional data. When we go for the anatomy, we already can see there is default anatomy, and we can check out what this default anatomy is. And then there is a MR, that is an average MR. It's a specific, very well-defined brain, that is a standard brain, and that is already included in the toolbox. And what is nice, there is from this MR already reconstructed the cortical surface. You can see here, this is just the cortical surface of the gray matter. And then there is also the inner skull, the outer skull, and the head. And you already, hopefully you can guess where this is going now, because we have this model already done, we know where the brain is in comparison to the head. So what is missing are the electrodes. Meaning this is all coming with the toolbox. There is no specific information yet, but we're going to add it. So all this anatomical information is already there. So what we can do is make a new subject. So here is our study. We just make a new subject that is called UT64 in our case. We do the same. We want to have default anatomy in one channel per subject. It's safe. And then we have a subject here in common files. There are no common files yet. What we want to have is now the channel locations. So we can just right click on these common files, import the channel file, and then we can have this folder, meaning in my case, it's Twin Brain Workshop, folder 5, BP64. Then here is the IC, the pre-processed and IC set you provided kindly with me. And you just hit, you have to select the CEG lab set, you just open it, and there's something loading, hopefully. It says there's a transformation. For now, we want to really do it from scratch because I want to demonstrate how this is done, so I click no. And now what we have here are channels. And those channels are not matching because they are template channels, they are spherical ones, but it's already somehow in the same space. You can see there is a template and the channels, but they don't match. And now the trick is to align those dots. Meaning we click right here, click on MR registration, and we want to edit it. And what we want to do is trying to align the template with the data coming from the EG lab set file. Meaning to have a better orientation, we click here on labels to have the labels. And then we know, okay, it's already too low for sure. So we can shift. We can click on this panel here and shift it upwards. I do that with my right mouse, shift it up. And now this is here the guessing starts. That's somehow somehow undefined part. If you have a picture or some idea where the electrodes were in your subject, it's already good. But here we have to guess because it's both default anatomy and default channel locations. That's why we have to do some kind of a qualified guess, let's say. So already it's high enough, but we have to stretch and bend them. So I'm just trying to do my best in stretching, bending, and shifting to get this these two spaces aligned. I think it's also a bit, yes, it's a bit, in the X direction, it's too little. Then I can shift, and what is important there, the standard views you can change with your numbers, meaning if you do 1, you have a specific one, and you can just go through and change the vision, and just even if it's a bit virtually setting up a cap, as if you would do it in your real world. You just try to do your best to make it magic, meaning you again squeeze and maybe make it a bit wider. And also the top view is always very important, meaning you have CZ a bit before the X-axis coming up. But this is already quite important. What is important to check is this axis is parallel to the X-axis. There is no shift, no tilt in either direction. So you can just check that through here. And this should be also symmetric, so to say, but this is already all right. And if you have something that is already quite okay, you can then further on project the electrodes on the surface, but you should do that only in the very end, that you have exactly all electrodes being exactly matching the surface. So if they're somewhere below or if they are in the air, that's not good, but you should have those guys on the surface. And then you hit OK. That will make a change, but then we can look at those guys again, and you can see now it's exactly what we want. There is a cap that is matching the cap we were using, and those guys aligned to the surface of your model. And you can also see that is somehow, you already have the geometry sorted out. Good, good. So far, so good. What you can do now is having this model, you can then use this template and electrode positions and compute the head model. And here we have to check all the referential points, but for now they're good. So those points are specified points defining the coordinate system. They are predefined, they're all good. And we can now select if you want to have a cortical surface reconstruction or volumetric one. For now, we go with the cortical one. And this is a bit now a delicate setting here, because this sets the conductivity values relative to each other. Meaning we have, we're going to set the conductivities relative to each other. And the most important factor here is this skull to brain ratio. And traditionally, this is set to 1:80. That gives okay results, but there are plenty papers showing this is rather 1:25. That's why I'm setting here to 0.04. That's also my suggestion. What to do. Then you click OK, and this is then running like 10 minutes. I won't spend 10 minutes in waiting now. That's why I prepared something. It's exactly the same thing here, but here in common files, I do not have only the. So let's do this. Let's erase the things I didn't explain. Let's say I did this, it was running 10 minutes, and then just have the channels and the open MEG bed. We also can look at those guys and look in the contents. And what we show here is we have a gain of 65, that is the number of channel, times 45,000. That is just 15,000 vertices times 3, because we have 3 dipoles. And if you even look at the cortex before, this is explaining this 50,000. This is then just the rendering of this cortex, because in the computer, everything is digital. And that's already a rendering of the cortical surface. And at every intersection here, there is one node, meaning at every intersection, there is one source. Those are 15,000, because we have 3D, it's 15,000 times 3 blocks. Also, it says cortex 15,000. If you do not have a super good fast computer, you also can downsample that to at about 5,000. I think that's still okay. But for now, we have this model describing the propagation of the potentials from the brain to the skull. And that we can use to make, this is called the forward model. This we can use to compute the inverse model. And this is very simple. You do again, right click in the toolbox, hit compute sources, and now it says there's no covariance matrix. That's exactly right. Covariance matrix modeling is important. And you can, in a very simple way, you can just compute the covariance matrix saying CC equals, oh, I have to change this. I'm not sure. Yes, you can just do this, you know, cove. In a very simple way, you could do this. You should not do it as simple as this because it's very important. Let's say that. Oh, sorry, I have the wrong keyboard here. So Swiss or German, I'm not so sure. Huh? Yes. Oh, yeah. So you should, the covariance matrix is very important. You only should use data from your baseline. In our case, would be standing the baseline. But I didn't prepare that, but it's important to consider a covariance model. For now and for the sake of time, I think I just use no covariance matrix. And then, like if you have a covariance matrix computed, you can import from MATLAB. And there might hopefully be a variable called CC somewhere. Hope yes here. But for now, I don't load it. But you should model your covariance matrix that is somehow the noise condition. It comes a bit from MEG, where people having a bias only in the empty room, and they usually use empty room noise measurements for computing these noise covariance matrices. In EG, we have to define a baseline that doesn't contain any signal of interest. That is a bit hard in EG. That's why it makes it more difficult. Oh, okay, now I have an effect I didn't expect. Let's see. I hope this doesn't take too long. I think, yeah, maybe I did something not too smart here. Let's give it a minute. And I was just start from scratch. So, all right. So if you don't do any noise covariance modeling for now, you always should do it. But now you don't. There's just the identity matrix, and then you can hit here. Compute sources. It's all very complex, but it's very handily integrated in the toolbox. It's a big achievement that this toolbox does it for you and even provides it with some presets. And now there are a lot of settings. So if you want to keep it simple, you hide the details. You don't care about it. But you should always check or at least report what's in your settings. And what I like, because I'm researching Switzerland, I like Loretta. There's Loretta. Because it's smooth and it's somehow, it's a version of what was developed in OLAP, that is Loretta, and as Loretta is a more modern version. And what I also like is not to constrain sources normal to the cortex. What is one approach that is well justified because we know the pyramidal neurons are parallel to the cortical folding, but I still like to have some variability in the dipole orientation. That's why I like to have loose constraints. So I can suggest using the settings as Loretta and loose constraints. The regularization is all right for now. So I just click OK, and this is done. This is very fast because somehow computationally, the difficult part is to select these constraints. But as soon as you decided for these constraints, and because those guys were implementing these solutions already quite elegantly, it's just so quick, and we have an inversion kernel that inverts the relationship of the forward model that we call the inversion kernel. Having that, also I pretend this was never happening. We make a new, let's see here, a new folder. What is a condition? And that could be just called Gait phase life in our case, and it's just an empty folder. Now I can do a right click, import EG, and then I can go, I go here and import from MATLAB because it's MATLAB variables I was writing. and I click on these MRCPs. And this is corresponding to what I showed you here. In the script I was writing MATLAB variable called mrcp.f final comment. And that's why it should be called like this. And that's exactly what's happening. It's called MRCP gate trace related because I was setting it up. And it's also important to check out what's going on in data. And as I said before, this thing is not only physiological, and I think also those guys are artifacts, and how to get an idea about that is to look at the data. So what I have here, is the timing and these huge fluctuations. If we look at these huge fluctuations, we can see a very strong activity in the back, as well as they are here. And we also can scroll through time, right? We can use the cursor. I'm using my cursor here to scroll through time, and that's very quite informative. If I have something fishy, then I have something that looks interesting to me. I have something that looks like an artifact, then I have something that looks interesting. Because this seems to be central activity again. Meaning what I want to do with this example is raising some awareness, again, looking at your results and then trying to find out if that makes sense or if it might be a result of insufficient cleaning. And because we did already the source imaging procedure, what comes with it is not only the topography, but also the sources. That's quite nice, I think. Because if I look at those guys, what I think they are artifacts, there's always a blob in the brain. That's the bad news. But what is the good news is somehow you can assess where this is mapping. And that's somehow mapping everywhere. It's really spread out, somehow very unspecific. But if you go a bit further on, you can then have the time course, the topography, and then you can serve a bit further. What I said, okay, this already looks a bit more interesting here. That seemed to be more isolated, right? Nothing else is active in the brain here. There might be this either prefrontal or eye artifact, I don't know by now. But this, to me, looks physiological, this negativity over central areas. But to really prove that, we have to look at more subjects. But it's just to highlight that if you just compute simple averages of your data, which are aligned to the artifacts, more or less, to the temporal structure of the artifacts, you might get, even after removing some ICs, you might get even some artifacts in, and you never can be sure that this is physiological. But here, if you just look at everything, it's somehow all over the place. It's unfortunately also somewhere else, but it's all spread out. Those guys, for instance, they're more distinct in space, and that's why I would say probably because this is so central and so isolated, that might be physiological, while the other would not. That's the first example. I just do a second example where I'm not only loading these MRCPs, but I'm also loading from MATLAB the IC maps. That's very similar what we did in EG lab already. I'm closing all those guys because this is what you learned already. You know about that. And that's very simple. This is just we can look again at the time. It doesn't make much sense because there is no time structure, but we can look at the maps. We can look at the disks. That must be exactly the same as we see in EG lab. It's just a different visualization. So for now, okay, let's keep the timing, but just for browsing around. And this is the first map, the second, and so on. Those are just the IC patterns, right? As you have it exactly in the same order and the same version you have it in EG lab. But the nice thing now is that you can do source imaging with it, meaning you have these IC patterns, and because those guys are linear, it's valid to look at those guys at distributed sources of the ICs. And what you can see here, this is also, some kind of a fishy result, because we know those are eye artifacts here, but there is always a blob in the brain. So what I want to question is, because you always have a blob in the brain, you not always should say, okay, this is now, olfactory activity, some kind of prefrontal ones. But you always get some source imaging result. You have to assess if this makes sense, if this is plausible. But what is nice, in addition to dipolar analysis, in cases you not only know that you have one dipole, especially in artifacts, you have some kind of source representation of your IC patterns that just come right away, right? This is just, I think, a very nice representation. And I think for that, I think I gave you a rough idea how to do that. How to use that in Gait is a bit more complicated because in frequency analysis, you have to image the real and imaginary part of your FFT results separately and then compute the magnitude in the source space. That's why I'm focusing on these linear relationships because they're easier to visualize. But for now, this is exactly the principle. We have some configuration and all linear transformations you do from your data, you're still able and it's valid to source localize. If you do non-linear transformations, these relationships are violated, meaning if you would have some kind of power topography and you try to do that trick here, that's not correct, because then the mathematical equations are violated. With that, I want to close and maybe discuss some questions, if there are any.